During gastrulation, a strip of epithelial cells on the ventral surface of Drosophila embryos undergo apical constriction, shrinking their apical ends to become cone-shaped rather than columnar. The simultaneous contraction of these cells creates a long furrow in the tissue that invaginates to form the embryonic mesoderm. In 2009, Adam Martin, a postdoc in Eric Wieschausser's lab at Princeton University, looked into the mechanism of this morphogenetic movement.
What we found was that the myosin appeared to be undergoing these pulses of contractions, and it constricted these cells in this ratchet-like manner. The focus of that paper and the focus that I had up to that point was really looking at the mechanism of constriction in an individual cell. But of course, this process is a tissue process, so you have over 1,000 cells undergoing contractile behaviors and generating this furrow. So we wanted to understand how forces are transmitted from the cellular level to the tissue.
An important feature of the process is that apical constriction is polarized or anisotropic. The cells primarily constrict in one direction, so that they lengthen along the embryo's anterior-posterior axis, forming a narrow ventral furrow.
If the cells constricted isotropically, what you get is a pit, not a long, narrow furrow. How does this anisotropy arise? It could be due to some sort of mechanical tension in the tissue. Maybe you have higher tension along the anterior-posterior axis.
Alternatively, the contractile pulses in individual cells might only constrict the cells in one direction. To see if this was the case, Martin used software developed by his collaborator Matthias Kashuba to measure how the constricting cell's anisotropy correlated with pulses of contraction.
If the contraction pulses are directional, you might only see the change in the anisotropy during a contraction pulse. There didn't really seem to be a significant correlation. So this argued against a model in which individual cell behaviors lead to this anisotropy, and supported the idea that the mechanics of the tissue is playing a role in determining the cell shape.
Tissue forces might be integrated through adherens junctions, which Martin's previous work suggested were pulled on by the pulsed contractions of the actomyosin cytoskeleton. Now he looked at the dynamics of GFP-labelled myosin in Drosophila embryos with a partial loss of armadillo, the fly homologue of the critical junction component beta-catenin.
When we imaged these embryos, we saw really a quite striking phenotype, which is that you see this meshwork of myosin assemble on the surface, and then you see these dramatic tears in the meshwork. And what was really striking was that the separation always occurred along the anterior-posterior axis, which suggested to us that the tension was highest along this axis.
To confirm this, Martin used lasers to sever the actomyosin network and measure the recoil in the surrounding tissue, which would reveal how much tension was built up in different directions.
We were able to collaborate with Rodrigo Fernandez-Gonzalez, who's in Jennifer Zollin's lab at Memorial Sloan Kettering. They have a laser ablation system set up, and I made different types of laser incisions across the surface of the ventral furrow tissue. If we measure the initial displacement of myosin structures around the cut, we see that there's 2 to 3 times more displacement along the anterior-posterior axis than the dorsal-ventral axis.
This again suggested that tension is greater along the anterior-posterior axis of the embryos. But does this tissue tension feed back to control cell shape by restricting the apical constriction of individual furrow cells? Returning to embryos that partially lacked armadillo, Martin found that after the epithelial tears opened up to release tissue tension, myosin continued to constrict the cells bordering the tears.
What was striking is that they constrict in a perfectly isotropic manner. It appeared that the cell wants to constrict isotropically and is generating force in all directions, but the mechanics of the tissue prevent the cell from constricting in one dimension, which is, of course, important for the formation of a furrow.
Tissue tension is probably generated by a supracellular meshwork of actomyosin fibers. Martin and colleagues found that the fibers of individual cells connected together at spot-like adherens junctions. When cell adhesion is lost in the absence of armadillo, myosin flows continuously into a ball in the center of each cell, suggesting that contraction is unrestrained and that adherens junctions normally balance the contractile forces in neighbouring cells to transmit tension throughout the tissue. The researchers then took a closer look at what happens when this balance of forces is disrupted.
Eric prepared some of these arm germline clones for electron microscopy. It really beautifully illustrated these membrane tubules or tethers that appeared to still be connecting the cells even after all the myosin had contracted into the middle of the cell. So what we think is going on is that cells lose mechanical connection to each other when we disrupt the adherens junctions. However, the plasma membranes do remain connected in some way, such that when one cell contracts away, it will pull out a membrane tether from its neighboring cell.
So spot adherens junctions connect up the actomyosin network and balance its contractile forces. But how are these forces regulated? Martin knew from his previous work that the ratchet-like constriction of individual ventral cells is controlled by two transcription factors, named snail and twist. In the absence of twist, pulses of contraction still occur, but the cell constrictions aren't stabilised, so the cells stretch back out in between contractions. To see whether pulsatile or stabilising contractility is required to generate tissue-wide tension, Martin knocked down twist at the same time as armadillo.
The cells still lose adhesion, however, you no longer get these tissue-wide tears, so there's much less tension across the tissue. So our model is that in order to transmit forces, you need all the cells to essentially be generating tension at the same time, because if one cell pulses but its neighbor is relaxed, then the forces are simply going to be dissipated because you won't be able to propagate forces to the third or fourth cell in the sequence.
So how does twist maintain tension in between contractile pulses?
We had noticed in wild-type embryos, the myosin on the apical surface of the cell is increasing over the time course of invagination. And so we wanted to look at how myosin dynamics were after knocking down twist through our collaboration with Matthias Kashuba and also a really talented undergraduate, Michael Gelbart. And what we found is that the myosin intensity increases during the pulse, but then drops back down to near basal levels.
So twist stabilizes myosin on the apical surface to maintain cellular tension in between contractile pulses. This myosin network is connected up via spotted adherens junctions to propagate tension across the epithelial sheet. This tissue-wide tension is anisotropic. It's stronger along the embryo's anterior-posterior axis, which feeds back to restrict changes in individual cell shape.
The distribution of forces across the tissue is critical for the outcome of tissue morphogenesis. We're trying to figure out how this anisotropy arises, whether the anisotropy is a result of the geometry of the embryo or whether this has to do with the expression pattern of snail and twist. And of course, we can address this using Drosophila genetics and looking at mutants that alter the expression of snail and twist in the embryo.
You can read more about the integration of contractile forces during tissue invagination in the paper by Martin et al., which you can find in the March 8th edition of the Journal of Cell Biology.